Today's notes are on mitosis and the cell cycle. The cell cycle is a series of steps that a cell goes through during its life cycle. It has three main stages. The first part is interphase. As you can see, the cell spends most of its time in the interphase stage. In interphase, cell is going to grow, and the DNA is going to be replicated, and then the cell will continue to grow until it's ready for division. Mitosis is the second stage, where the nucleus is going to divide, and the third stage is cytokinesis, where the cell is going to divide. So in a cell's journey, which is a long cycle, it's going to grow. The DNA will be replicated so that when the cell splits in mitosis, there will be DNA ready for both cells. So in your notes, what I'd like you to do is summarize what happens during the cell cycle. Interphase is the first stage we're going to talk about. It is the longest phase in the cell cycle, and the cell spends most of its time in this phase. And we're going to talk about the three different parts of interphase on the next slide, but take a look at this image here. This is an onion root tip. Onion root tips are plant cells, and we know that because of the box-like shape of the cell, right? That means that they have a cell wall. This red circle represents the nucleus, and that little tiny red, very dark red circle represents the nucleolus. Okay? What happens in interphase is the cell is going to grow, and that's going to happen in G1 phase, growth phase. And that's going to be the normal functions for the cell that are occurring. So if it is a muscle cell, it's going to be performing a lot of cellular respiration in order to produce energy for those muscles. If it's a neuron cell, it's going to be sending chemical impulses to and from the brain. And then once this cell has grown to a specific point, S phase will occur. S phase stands for synthesize, which means the DNA is going to be copied. Synthesize means to make. So we're making a second set of the DNA. And this is very important because when a cell divides, each nucleus needs to have its own set of DNA that matches the cell that was there before. And then after the DNA is replicated, the cell enters G2 stage. G means growth. It continues to grow and prepares for cell division. In mitosis, we have a picture here of onion root tip, and you can tell that most of the cells, cells are the box-like shape. The large circle is the nucleus, and the inside, the dark circles, are the nucleolus. And you can tell that most of the cells are actually in interphase. Okay, most of the cells are in interphase, but however, some cells look a little different. Okay, some cells look a little different, and those are specifically the cells that are undergoing one of the stages of mitosis. And we're going to talk about those stages here. The first stage of mitosis we're going to talk about is prophase, and this is where the DNA coils into chromosomes. So you can see here in this image, there look like little X's here. Those are chromosomes. The nuclear membrane starts to disappear. So what we saw before the nucleus, the nuclear membrane is just like the cell membrane, but instead of being the outside of the cell, it's the outside of the nucleus, and it's starting to disappear. And if you look outside these orange things, these are the centrioles. And these centrioles create spindle fibers. And the spindle fibers are very important because they're going to help move things in the cell. So you can see prophase, right, looks like this. It looks like a regular cell, but instead of having dark circles like this with the nucleolus inside, you're able to see the chromosomes a little bit more clearly, and the nuclear membrane is definitely disappearing. A good way to remember prophase is P is for prep. So this is our prep stage. We're getting ready for mitosis. We're getting ready for the cell to divide. Metaphase is another simple one. Chromosomes line up at the center or the equator of the cell. So you can see here that the chromosomes line up along the middle or the equatorial plate. You can see the centrioles at each end of the cell, or what we call the poles of the cell. The spindle fibers are attached to the chromosomes on each side. So in an animal cell, and we know this is animal cell because it's circular, you can see all the chromosomes are aligned in the center. In the plant cell, it looks very similar. Again, the chromosomes are lined up in the center. An easy way to remember this is M is for middle. Chromosomes line up in the middle. Anaphase is when the double chromosomes separate and move away to opposite ends of the pole. So you can see here, these big X's were once attached, and now they're being pulled to the different sides of the cell by the spindle fibers. Okay, here's what it looks like in a plant cell. You can see it looks kind of interesting here. We have the chromosomes on this side going over here, and these halves going to the other side. Here's what it looks like in an animal cell. An easy way to remember anaphase is A for away. The chromosomes are moving away. Telophase is the last phase in mitosis, and this is when the DNA starts to uncoil, the nuclear membrane starts to reform, and the spindle fibers are broken down. So you can see we can't really see any more spindle fibers. Now you notice there is still only one cell. So an easy way to remember this is telophase is for T for 2 nuclei. There are now 2 nuclei in the cell. And this is very important because the cell has not yet divided, which means the question is, when does a cell divide? Well, it's going to divide in cytokinesis. Cytokinesis is the last stage of the cell cycle, and this is really important because the cell divides into 2 cells at this point. So cyto means cell, kinesis means to move, so the cell is moving, and this is the division of the cytoplasm. There are differences between plants and animal cells here. In a plant cell, a cell forms a cell plate in the middle, and what happens is the cell will just split up and separate just like that. Well, an animal cell is going to pinch to form 2 cells, or what we call a cleavage furrow. So you can see that the cell starts to pinch from both sides, and eventually 2 cells are formed. So the question is, how do we remember mitosis? Well, a good way to do that is P-MAT. Okay, P for prep, M for middle, A for away, T for 2 nuclei. So P is prophase. This is when the cell is going to prep for mitosis, so the chromosomes start to form, the nuclear membrane starts to disappear. M is for metaphase, or the middle, so chromosomes line up in the middle. A is for away, so chromosomes move away to opposite ends of the cell. And T is for 2 nuclei, so 2 new nuclei have been produced. So let's take a look at the cell cycle one more time. The important thing to remember is that this is a continuing cycle. So as soon as the 2 cells are divided, they're going to enter interphase again. So it's a continuing cycle. So first we have interphase, where it's going to grow and DNA replication will occur. Then mitosis will occur, where prophase, metaphase, anaphase, and telophase occur. And then the final step is cytokinesis, where the cell becomes 2 new daughter cells. What I'd like you to do is complete the summary and questions.